Good afternoon, respected sir. It's me, Isha Thakur, from class BSc Biotechnology first semester, roll number 23012 from Hansraj Mahila Mahavidyalaya. Today I am here to present my presentation on the topic microbial growth. Further, for the acknowledgement, I would like to express my heartiest thanks to our worthy professor Jatinder sir for providing the opportunity to express our understanding regarding the topic. In the index, we have the introduction, how to measure growth, measuring optical density, study of growth, phases of growth, generation of time, growth characteristics, factors affecting growth. First of all, we would start with growth. What actually growth is, with the definition of growth. It is defined as orderly increase in the chemical constituent of the organisms. Some of the key features of growth are refers here, like it refers to an increase in cell number. Each phase has specific length. It is integral part of every living organism. It is symmetrical process and it cannot be reversed. Further, we have how to measure growth. We know what is growth, but then how we can measure growth? Yes, it can be measured. It can be measured by determining cell mass or the cell density, or by counting number of cells. Determination of OD can also be done, that is the optical density. To measure the optical density, we have an instrument known as spectrophotometer, which is defined as an analytical instrument used to quantify measures the transmission of reflections of visible light, UV light or infrared light. This is how the machine really looks alike. Next, we have, why do we study growth of the microorganisms? So, it is necessary that when we are aiming to utilize or inoculate known number of bacterial isolates, then we need to study the growth. For example, enhance plant growth, increasing biodegradation of toxic organics, produces antibiotics and others. Further, we have phases of growth. Now we have different phases of growth. First is lag phase, second is log or exponential phase, third is stationary phase, fourth is death phase. First of all, we will discuss about lag phase. For long time, lag phase lasts for 1 to 4 hours. Bacteria adapt themselves to growth conduction. It is a period where the individual bacteria are maturing and not yet able to divide during the lag phase. Next, we have our log phase, that is the exponential phase. In this, rate of growth is constant. Population number of cells undergoing binary fusion doubles at constant interval called the generation time. And this continues as long as cell have adequate nutrient and good environment. Next, we have the stationary phase, which actually lasts for few hours to few days. It can be defined as stationary phase is due to a growth limiting factor. This is mostly depletion of nutrient and or formation of inhibitory products such as the organic acids. Next, we have the decline phase or the death phase. It is the nutrient deprivation phase. Waste material is accumulation, irreversible loss of the ability to reproduce. The death can be logarithmic and can vary with the environmental conditions. Resistant bacteria shows extended survival. General bacteria dies in the log phase. Generally, a few of the bacteria just dies in the log phase. Now, we will study about generation time. It is also known as doubling time of bacteria. In most of the bacteria reproduce by binary fission which take place specific duration, that is called the generation time. Here we can see the E. coli. Next we have the mathematical expression for our growth outcome. And on the last we will find that log 2 is divided by log bn by log bn. Further, we have relationship of generations with time and growth. This is the graph showing the relationship of generation time and growth. Next we have the growth characteristics. Each bacterium when grows in a medium shows specific growth appearance or characteristics. These specific characters are generally useful in the identification of known or unknown culture of bacteria. Factors affecting growth of microorganisms. Mainly the physical factors affects the growth of a microorganism. So in that we have solute and water acidity, temperature, pH, oxygen requirement, pressure and radiation. Next, we will discuss one by one all the physical properties that lead to this. Solutes and water acidity. If the cell cytoplasm possess higher solute concentration to comparison to its environment, then water diffuses and after this the osmosis take places. Further from the osmosis plasmolysis take place, and here we can see different types of surviving or non-surviving organisms. Temperature. Yes, temperature is the most crucial factor that affects the temperature that affect the growth of an organism. Like enzyme have a range of thermal stability and beyond it their duration take place. Thus high temperature kill microorganisms by denaturing microbes. Shows characteristic temperature depends and possess its own critical temperatures. Next, we are going to discuss about the pH, how pH also affect the growth. Such like most bacteria grows best around neutral pH values, like from ranges from 6.5 to 7.0, but some thrive in acidic condition and some can even tolerate a pH as low as 1.0. Next, what is the minimum? We have this a table which shows the maximum and minimum pH of growth of some species of microorganisms. Next is the oxygen requirement. As we know, oxygen is a very vital source for all the living organisms to survive. So in our environment, oxygen content is about 20%, and the organisms or the microbes that depend on oxygen are called the aerobes. Then the organisms that can be present in the absence of atmospheric oxygen, they are known as anaerobes, and the microbes that completely depends on oxygen for growth are obligate aerobes. Further, we are going to discuss about pressure. Normal life of microorganism on land or in the water surface is always subjected to as a pressure of the atmosphere. Some microbes are really very resistant to atmospheric pressure, so they can survive in even high pressures. Then we are talking about radiations. Some electromagnetic radiations are very harmful for the bacterial growth, especially ionizing radiations, for example, the X-rays or the gamma rays. So with this, we are going to end with all our topic. Thank you so much for listening till this point. Thank you. Hope you liked my presentation.